This lecture is being brought to you in part by the generous gifts of these sponsors.
Thank you very much, Ken, for that introduction, and thank you very much for coming this way. I understand that it is warm, so I will try to keep this as exciting as possible to keep you awake. So I'm, as mentioned, I'm at the Laufer Center for Physical and Quantitative Biology at Stony Brook and also at the Martino Center for Biomedical Imaging at Harvard MGH. And what I have been working on really is developing neuroimaging tools. And neuroimaging tools are specifically optimized for imaging in humans. And when they were first, starting to be developed about 20 years ago, the National Institutes of Health really viewed neuroimaging as paving the way for doing for psychiatry and neurology what, say, the ECG was meant to do for cardiovascular medicine. So instead of visiting a psychiatrist and sitting on a couch and talking about your troubles and the psychiatrist trying to figure out what might be the issue, the idea is you put someone in the scanner and you would get an answer as to what the actual diagnosis was, what the severity was, what kind of medication would be the appropriate medication, what the dose should be. In fact, that has not happened, and I'm going to try to talk to you about why I think that is and what the path forward should be. So let me give you a brief introduction to some standard methods of neuroimaging. So this one over here, this would be structural MRI. This is probably something you've seen before. This provides sort of the landscape for what the brain looks like. Of course, it can be applied to other parts of the body as well. There's a method called positron emission tomography, which can be used for lots of different purposes, but in this context, it can look at energy usage within the brain. So when the brain is using glucose, for example, blood sugar, it shows exactly how much is being used and where. There's a technique called MRS, or MR spectroscopy, that can identify different metabolites in the brain, different amounts. Functional MRI. Functional MRI is somewhat like structural MRI, except instead of looking at the landscape, you're really looking at the action. So you have someone in the scanner doing something, and then you're seeing how the brain is responding to what you're doing. Diffusion tensor imaging is a form of structural MRI that is actually showing the connectivity, the parts of the brain that are actually attached to other parts of the brain by white matter tracks. Here, this is a functional connectivity map, which is doing functionally what this is doing structurally. So again, when you're thinking hard, it's actually determining who's talking to whom. And my lab has been very involved in the beginnings of the development of a technology called near-infrared spectroscopy, which is an attempt to essentially take MRI-type imaging into ambulatory environments, like emergency rooms, rural environments, military bases, and so forth. Now, all of these techniques have certainly their strengths, and they give us valuable information. And yet, in some fundamental ways, these techniques are also incomplete. They fail to tell us, in some ways, what we most wanted to know from our neuroimaging experiments. So, for example, we want to know, will patient A recover, degenerate, relapse? What is his or her individual time course? What will happen if we introduce X or Y intervention? So when you look at how neuroimaging studies are typically set up, they involve large groups. For example, you have 40 subjects of one type and 40 subjects of another type, and you do a statistical analysis and you ask, are there differences between those groups? Or you have people under one condition and another condition and then ask, are there differences between those conditions? But when you go to a doctor's office, you're one person, you're not 40. And so a lot of these methods were optimized for groups, whereas if we want to move towards individualized medicine, what we need is actually a method that works for a single individual. But anyway, actually, I would say the difference is even more kind of philosophical and fundamental. Because yes, we can optimize the signal-to-noise ratios to be able to get better signals that can be detectable at the level of a single individual. But ultimately, if you look at these different neuroimaging methods, what you see is a map. You see an activation map where there's a picture with a bunch of blobs. So this way of thinking about medicine is really based on an injury-based model, which came out of neurology. Neurology was originally very focused on lesions. So you have a part of your brain that's missing or that has a tumor, and you want to find out where it is, what it looks like, so that a surgeon can take it out. But there's a fundamental difference between an injury and a disease, right? An injury is something that happens all at once. You are, standing in a road, a truck runs you over, all of a sudden you were in one state, now you're in another. That's not the way a disease works. A disease process is something that happens very, very gradually. And fundamentally, a disease process is actually a breakdown of the regulatory mechanisms that maintain homeostasis in the body. So what do I mean by that? So in our bodies, we have lots of values that have to be maintained at a specific set point. Those values are a problem if they are either too high or too low, right? And so what the body has to do is to maintain them, within those set points, regardless of chaotic inputs from the environment. So here's an example of the metabolic control system. If I eat a donut or a cookie, my blood glucose will go up, but it's dangerous for my glucose to go up too high. And so that will trigger a response of insulin. My insulin levels will push my blood glucose down. And if my blood glucose is too far down, then glucagon will push my blood glucose up from glycogen, converting glycogen to glucose. And so the body is actually optimized to maintain homeostasis, and disease is the process that occurs when that homeostatic regulation starts to break down. So ultimately, what we want to do is to be able to say something about a trajectory. We want to ask, if we want to in fact use neuroimaging to say something about personalized medicine in the context of disease, because all of these up here are diseases. So here we want to ask, is a child likely to develop some psychiatric illness? Is a person likely to have another seizure, and if so, when? If you have an addict, is he going to be able to fight the addiction? And if so, what is the likelihood that he's going to relapse? And if so, when? And of course, there's always the fact of changes that occur just as a natural part of aging. So what we'd like to know is, what is the time course? So if there's a response, what is the time course with which it comes back down? Is it, a flat time course? Is it something that goes up dramatically and then down, and so forth? And none of those imaging methods, as they currently stand, are designed to do that. So what we're really looking at is something called a control circuit. A control circuit has excitatory and inhibitory components that maintain homeostasis in the face of chaotic inputs. So what does that mean? This room is somewhat warm, right? So when you have a room with a thermostat, when the temperature goes up, that triggers the thermostat to turn on the air conditioning. That causes the air conditioning to turn on, which lowers the temperature, right? So that's a working regulatory circuit. These circuits are ubiquitous within engineering, of course, but they're also ubiquitous within our bodies. So if we had to say what is different about disease processes that we want to capture with our neuroimaging methods, there are three main take-home points here. One is that what we're looking at is dynamics, not higher or lower values, right? So if we want to know if the thermostat is working properly, it's not enough to ask what the temperature is. We need to know what the time course of the temperature is. The dysregulation is seen long before symptoms. So in other words, the dysregulation is not something that, like an injury, happens all at once. It's something that happens slowly and becomes more and more serious over time. That's actually good news for us in terms of medicine, because it means that we might be able to catch a dysregulation when it's still very mild, treat it before people become symptomatic. The third point is that dysregulation, even of the same circuit, isn't one thing. It's many. There are many ways that you can be dysregulated. In diabetes, for example, you have type 1 and type 2 diabetes. One is a dysregulation of the feed-forward part of this control circuit. The other is a dysregulation of the feedback component of this control circuit. Why does this matter? Well, if you look at neuroimaging results in psychiatry, almost every psychiatric disorder implicates the same regions of the brain. And yet, instinctively, we know that anxiety feels and looks different than depression, which feels and looks different than schizophrenia. So if we want to say something meaningful about what a psychiatric disease is, we are going to need to go beyond asking which parts of the brain are involved to be able to say something about their regulation, because they're going to be dysregulated in different ways. And that's what we're trying to do here. So the introduction to this work came from the Office of Naval Research, which had a particular problem. And their particular issue was that they were interested in developing a neurobiological diagnostic for Navy SEALs and other special forces. And just as they went through training that assessed, physical strength and ability to withstand lack of sleep and lack of food, they wanted to be able to develop a neurodiagnostic that would show the brain's ability to withstand stress. And the idea was that they didn't want to find out, once they dropped someone behind enemy lines, how that person was going to respond to an extreme stressor. They wanted to be able to know ahead of time so that they could pick the right people, not only for physical resilience, but also for emotional resilience. So I thought, okay, that's an interesting problem. At the time, I had been working on schizophrenia, but I thought this is a way to get to a much more fundamental question and a way to address this problem, this circuit problem, in a much more fundamental way in a healthy population. And so I started working on it, and the place that I started was with this prefrontal-limbic circuit. This is the canonical circuit developed by this person, who's Joe Ledoux. He's at NYU, and he is really the person who started thinking about how does the brain process fear. So if a snake, or your angry squirrel in this case, drops out of the ceiling, initially, your sensory thalamus would register that something is happening, and it would send two signals. One is a short, quick response, excitatory, to the amygdala, which would make your heart rate go up and you'd start sweating and you breathe more heavily. But in parallel, it would send a longer signal to the prefrontal cortex, which would then suppress the response. So you wouldn't remain excited forever. You'd remain excited and then calm down. And that's what's happening here, where the excitatory response is pushing you up, the inhibitory response is pushing you down. Now, I want to point out that this is, these are a bunch of boxes connected by lines. Okay, that's not really how engineers think about circuits. This is how engineers think about circuits. Okay, and so what I started doing was going through all the animal literature and trying to identify exactly which neurotransmitters were connecting, which areas to which areas, and you wind up with this, which is absolutely a mess, right? I mean, nobody wants to try to solve this equation. But as it turns out, you don't need to, because in any circuit diagram like this, there are important parts of the circuit and less important parts of the circuit. And what you want to do is to try to simplify this to the point where you see who are the main players. So there are two ways to do this. The local way is to, in fact, identify what is the connection from each one of these boxes to each one of these boxes. These wires look like wires, but they're actually differential equations, and specifically ordinary differential equations. When you put them all together, it's a system of coupled differential equations. So why does that matter? So this is fundamentally different than those pictures of blobs, in the sense that if I put an input to this model, it'll give a response. This is a system that will allow you to put in any input that you like, and it will, because it's a model, it actually can predict what the output will do over time, right? Now, the problem with the local response is, even though you think you know something about the system because you know what's happening at each one of those wires, in fact, that's a very dangerous response, because it assumes that you have all the information. And I'm a very skeptical person by nature and suspicious even of my friends. And so I thought, you know what, I don't trust this. For one thing, this was done in mice, and I'm not a mouse, right? And I'm studying humans. So it's a bit dangerous to invest too much in this sort of circuit diagram. And so the problem is that if you guess incorrectly as to what these wires are, you create an equation that is completely incorrect. So we're going to start with something that's much more agnostic, and that's the global approach. And the global approach says, you know what, I might not actually know each one of these connections, but I know the behavior of a well-regulated control circuit versus one that isn't. And you do too. Because if their air conditioning thermostat is working properly, you know what the dynamics should be of the heat, right? So the heat should go up, the air conditioning should go up, the heat should go down, the air conditioning should go down. So in a well-regulated control circuit, the dynamics have a particular signature. And in a dysregulated control circuit, they have a different signature. So here we have an example of dynamics of a dysregulated control circuit. So we have here individuals with diabetes, and you can see that the dynamics of the healthy control, in red, are quite different than the person. There are two people who are type 1 diabetics. And so somehow we're going to need to quantify that, because that's going to give us some important information about how dysregulated the system is and where. So here's another example. Here's in heart disease, we have on the bottom, well, on the top we have a normal heart rate. So this is a heart rate that's changing in time. On the bottom we have someone with heart disease. And the dynamics look different. You can see that, right? You don't need fancy math. You can just see that the dynamics look different. So what's happening? In the top one, someone's heart rate is going up and then it goes down. It goes up and then it goes down. Why? Because all sorts of things happen to you during the day. And so if you have a well-regulated control circuit, your heart rate goes up and then it pushes it down again, right? In someone with heart disease, it goes up, but then because the parasympathetic nervous system is very sluggish, it takes a long time to bring it down. And you can see that in the dynamics. So we can quantify this using fractal analysis. So many of you may be familiar with spatial fractals. Fractals are mathematical structures that are similar across many different scales. So there's some pretty pictures on the internet if you want to check them out on fractals. But in fact, you can have fractals that are also not only spatial, but temporal. So what does it mean to be a temporal fractal? It means that the same patterns are visible at many different scales, not in space, but in time. So as it turns out, this can be quantified. So if all frequencies are equally well represented, we say that the beta have 0. This is white noise. That's what static is. It means all frequencies are equally represented. An equal balance between chaos and order would be pink noise. So total chaos is white noise. A balance between chaos and order is pink noise. And as we introduce more and more order, it gets to brown noise and eventually black noise. Now, why would physiological systems operate within a pink noise environment? Why would they want a balance between chaos and order? Well, because if you think about this is sort of an optimization strategy. So your body needs to be able to be supple enough that it responds to the environment. It needs to have enough flexibility that it can respond to the environment. But the system also needs to be constrained enough that it can return to baseline. Too chaotic, you'll never return to baseline. Too ordered, you'll never respond to the environment. And because the body is trying to optimize to require the least amount of energy to maintain homeostasis, it will naturally self-organize to pink noise. So that's a valuable tool. I mean, we also did some theoretical work that in fact shows that when excitatory and inhibitory components of negative feedback loops are perfectly balanced, this actually produces pink noise. And as you have a stronger excitatory or weaker inhibitory component, it's more chaotic. If there's a weaker excitatory or stronger inhibitory inputs, it's more constrained or less chaotic. So wow, you might say, why are you showing me all of this? Well, it turns out that, again, we can look at the dynamics, and just from the dynamics of the output, we can say something about how well regulated a control circuit is and why. Is it too constrained? Is it not enough constrained? So what part of the circuit seems to be broken? Again, why does this matter? Because for personalized medicine is looking at disease. Disease, we said, is dysregulation of a control circuit. So if we want to diagnose disease, we have to determine if the system is in fact dysregulated, and if so, what kind of dysregulation it is, because that'll tell us something about which disease it is. Now, the neuroimaging that people typically use, as I mentioned, is optimized really for large groups. Because I am working on neurodiagnostics of single subjects, of one person, we needed to radically increase the signal to noise. So to that end, we have been developing equipment actually to optimize acquisition parameters and increase signal to noise for neuroimaging. We also moved pretty much exclusively now to ultra-high field fMRI. So typically if you get an MRI, you'll get a 1.5 Tesla, maybe a 3 Tesla scan. We are currently using 7 Tesla for humans and are moving to 15 Tesla. And so what does that mean? It means that typically the signal to noise that you would get in a group of 16 to 25 individuals, we can get in a single individual. So in this particular case, we can perturb this limbic circuit by having someone be in a scanner, and then we're going to try to get that prefrontal limbic system to respond. So you can't really have a squirrel jump, fall from the ceiling in a scanner, but what we do instead is we have a countdown, and the countdown, either if it's blue means nothing is going to happen, or if it's red it means there's going to be a really unpleasant thing that happens at the end of that countdown. That could be an electric shock, it could be a loud aggressive sound like ah! And what will happen is that the prefrontal limbic system of that person will respond. And from that response, we can tell something about how the system is regulated. So here we can see that we're activating the parts of the limbic system that I showed you. So this shows that the task is actually working because it's activating the amygdala and the prefrontal cortex, which are the excitatory and inhibitory components of the circuit. Now, when we do our fractal analysis and compare it to trait anxiety, so some people are more anxious in life, some people are less anxious, right? And when we look at anxiety and ask, for the people who are more anxious, what is different? So what we find is that there is a dysregulation of the prefrontal limbic system in people who are at the more anxious end of the spectrum. And it's not random throughout the brain. It is, lo and behold, very specific to the prefrontal limbic system, exactly that circuit that I was telling you about that came from Joe Ledoux, with one exception. So Joe Ledoux has all of these different areas, but we found one that he didn't find, the inferior frontal gyrus, or BA45. The IFG actually was the strongest response, which was pretty interesting. So what about the other end of the spectrum? So now we have been looking at individuals who are on the more anxious end of the spectrum. What about people who are on the more stress-resilient end of the spectrum? It's easy to find people who are more or less anxious. You simply ask them, what sorts of environments, trigger you. For most people, they don't upset them. For you, that maybe they do. To ask whether someone's stress-resilient, you can't just ask them because they have to have something bad happen to them. And we're limited for reasons of ethics in terms of making bad things happen to them. So what we did is we actually designed an experiment where we had people spend 2 days in the hospital. And you had this session A where we take a bunch of blood samples and EKG. At the end of the day, we then do an MRI. And on the second day, we do exactly everything that we did on the first day, except we have someone jump out of a plane for the first time. You didn't see that coming. Neither did my department chair. So anyway, so while people are, going through this experience of getting ready, they're in the plane, they're going up to 13,000 feet, they jump at 13,000 feet, they're in free fall for a full minute, under the canopy for 4 minutes, and then they land and we follow them for 6 hours. They wear clothing embedded with biosensors so we can actually monitor all of their vital signs. So we're looking at heart rate and respiration and galvanic skin response and EEG and so forth. And so we know exactly what their response is to the stressor. And so we get something that looks like this curve for each individual. So you can see this curve over here. And this is basically a subtraction of those 2 days, right? So people have normalized for their own normal heart rates. And you can see where the skydive was, right? That's not very subtle. By taking the area under the curve, we can say how responsive someone was to this particular stressor. So as it turns out, the prefrontal limbic system, again, the regulation is very predictive of how people respond to this particular stressor of jumping out of a plane for the first time. The interesting thing is that the brain was quite predictive of how they responded to jumping out of a plane, but asking them all sorts of psychological assessments ahead of time was absolutely not predictive. So asking them, are you someone who typically gets stressed? Are you someone who is resilient? Are you someone who likes doing dangerous things? None of this was at all predictive. The only thing that was actually predictive, well, there were two. One was the brain, looking at the brain regulation, and the second one was their age. So both biological measures. So interestingly enough, again, the area that was most implicated was BA45, or the IFG, the inferior frontal gyrus. Now, for those of you who are paying attention, this was the same area that was dysregulated in people who were clinically anxious, or actually trait anxious, right? So how could that be? So you have people who are at the more anxious end of the spectrum, people who are trait anxious, who are showing dysregulation of the IFG. And then you have people who are responding least to the skydive, and they are also showing dysregulation of the IFG. So these people are at opposite ends of the spectrum. Why are they showing dysregulation of the same area of the brain? So this was actually incredibly puzzling to us. But if you look more closely, there was a really critical difference, because the IFG of people who were anxious was completely intact, whereas the IFG of people who were non-responsive to the skydive was actually degraded. There was atrophy. And so here's an example, as I said, with type 1 and type 2 diabetes, where you can have dysregulation of a control circuit in two different ways. And just as type 1 and type 2 diabetics look very different from one another, type 1 diabetics tend to be very skinny. Type 2 diabetics tend to be more overweight. In fact, in this case, both ends of the spectrum also show diametrically different symptoms. And in fact, this regulation of IFG, this fractal measure that I told you about, actually predicted how they rated faces, ambiguous friend versus foe types of assessments on their way up to jump. So in fact, people who were actually in the pink noise range were very accurate in discriminating between friend versus foe, between aggressive faces and neutral faces that were ambiguous. Whereas people who were non-responsive to the skydive and had dysregulation of the IFG also actually were not very accurate in assessing friend versus foe. They tended to see safety when in fact the faces were aggressive. So that made me think, well, maybe the reason they're not responding to the skydive is not that they are brave per se, but they're not seeing the danger, right? They are actually jumping out of a plane and their brains are not picking up on the fact that what they're doing is dangerous. And that made me think about the spectrum very differently, in ways that we'll get to in just a second. So here we have a test case for what I've just described to you. So we have Tim on the left. He's an EOD, which is explosive ordnance disposal. So he's someone who diffuses bombs for a living. And we have Dan on the right, who's a civilian. He's actually a television host. This was for a program we did for Discovery Channel. I don't know if any of you know Curiosity, but this was filmed for them. They wanted a demonstration. They'd heard that we were doing this stuff, and they wanted to see it for themselves. So they provided Dan, their host, and then we provided Tim. And in fact, so you can see these for approximate entropy, power spectrum scale invariance, Poincare maps. There are many different ways to describe this chaos versus order measure, but you can see the dynamics very clearly discriminate between these two individuals, even when we're just talking about two people. And most importantly, when we're not talking about a sick person and a healthy person, we're talking about two people who are healthy, who have different traits, different temperaments. Okay, so it's a very sensitive measure. So in fact, looking further, we can look at a diagnostic classification. We've developed statistical classification methods. And in fact, you can see that here we can discriminate. Here's generalized anxiety disorder versus control. So these are people who are anxious versus controls. We're able to discriminate with 90% accuracy. And now this is a more difficult one. This is people with anxiety plus depression versus people with anxiety alone. We were still able to distinguish them with 89% precision. Schizophrenia positive symptoms, schizophrenia negative symptoms, ALS, sleep versus wake. So we basically use these measures over and over and over again to test. These are not statistical comparisons where we have one group and another group and we're comparing them. This is where we have one person We take their measure, and then on the basis of that measurement, we decide you're in this group versus that group. And we're asking how accurate are you in making that discrimination? So it's a much more strict task. So thus far, we've been talking about insufficiently constrained systems. So we said that the system is closer to white noise. The system is too chaotic. So it's dysregulated in the sense that the system is too chaotic. There's not enough feedback. But the system, again, can be too constrained. So it's a mistake to say, oh, chaos is bad, order is good. That's not the case. The way physiology works is that a balance between chaos and order is necessary, and when you deviate in either direction, you get sick. So in this case, we're looking at an individual who's had a hemispherectomy. This is an individual who's had epileptic seizures, and they've occurred to such a degree that neurosurgeons have tried to take out the area that is producing the seizure, and in this case, they didn't know where it was. So in the absence of knowing where the seizure is coming from, they take out more and more and more of the brain until at this point, they've taken out almost half the brain. And let's hope they took out the right half. So here's an example. So this is a real problem, right? This is not a fantasy academic problem. This is a real problem where neurosurgeons simply don't know, how to identify focal points for seizures. So here, this is a problem that was brought to me by a neurosurgeon. This was a 24 individual male having upwards of 100 seizures a day. And we were asked, can you try to identify where this person's seizures are coming from, because we don't know where to cut. And bear in mind, this person's MRI looked perfectly normal. So there was nothing about the MRI that was giving any clues as to where to cut. And in fact, when we apply these complexity measures, as you can see, they're very clear points of singularity here that indicate a dynamic disturbance. And they show an overly constrained system. And so if you take those as seed regions, you can see that what's happening here is that those focal points are actually overly connected. So in the other case, they may not be sufficiently connected. In this case, they are overly connected, which might provide an explanation for how seizures propagate in other parts of the brain. So presumably, we all have little microseizures all the time, but they don't necessarily propagate. But as the structure of the brain changes and you have greater connectedness, this can potentially allow the brain an easier time to sort of propagate aberrant dynamics. So taking this now as a classification problem, here we have 19 individuals with left temporal lobe epilepsy. So in this case, we have a ground truth because they have specific symptoms that indicate where the seizure is coming from, and 19 matched controls. And then we wanted to test this method against that ground truth. Is this successful in identifying where the seizure is coming from? And this had 100% sensitivity and selectivity in distinguishing those individuals and in identifying where the seizure was coming from. Whoops. Okay, so now that we've taken a little break and said, okay, that the system could be too constrained or not sufficiently constrained, both lead to disease states, now we can go back to this original prefrontal limbic circuit, and now we're going to take individuals further at the end of the spectrum. We're going to now look at individuals who are clinically anxious. And by now, we're starting to learn a little bit, right? That's how science works. You do a series of experiments. Each one is teaching you something. And it's very exciting. You're chasing an idea and you're getting closer and closer. And of course, often your ideas are wrong. And being wrong is just as exciting or more so than being right because it leads you in a new direction. So one thing that we realized was that, that the IFG must have something to do with assessing ambiguous threat. And this must have been why Joe Ledoux didn't see it in his rats, because when he was doing these experiments with his rats, he had threat and non-threat. He had, electric shock versus not. Whereas what we had was something that was ambiguous, right? So could it be that what distinguishes people who are more anxious versus less anxious is not in assessing threat that's obvious, but in assessing the potential for threat? And so, in fact, that seemed to be the case, that controls are able to distinguish very clearly between threat and non-threat, and by that I mean not only in terms of their behavior, but also their brains, their prefrontal limbic systems. Whereas people with anxiety disorders, they tend to sort of treat everything as if it might be a potential threat. And in fact, at this point, the story is getting rather old. Again, we saw a dysregulation of this IFG. This troublesome IFG region that nobody had ever implicated in this control circuit kept reappearing over and over and over again in different studies, different design paradigms, different populations. We keep seeing this IFG. And this time we started now applying the local method. We talked about the global method. Now we're going back to the local method, and we're saying, okay, now we think we know what that circuit is, and so now we're going to start identifying what these differential equations are, these connections between different parts of the brain, so we can understand who's connected to whom. And what we found is that actually there were big differences between these two areas of the population in terms of not only functional connectivity, but also structural connectivity. And these functional and structural changes were also seen in children who were already showing some warning signs of susceptibility for later mental disorders. So this was done with near-infrared spectroscopy. These were 44 children, ages 3 to 5. Thus, the spectrum approach to stress resilience is what I would call a category error. It's an ill-posed problem. It's not the right way to think about it. It is not correct to say that on the one hand, these are the people who are stress vulnerable, and these are the people who are stress resilient. It's not that actually these are one thing. In fact, if you talk to people within the operational community, they are not interested in people who are fearless. People who are fearless get killed rather early. So what they want is someone who is actually in this range, in the pink noise range, who is able to identify friend versus foe accurately, who feels the fear but is not actually paralyzed by it, because the fear is actually an important piece of data. It tells you something about risk, right? And so what you want to do is to be accurate in your risk assessment. So if we actually bring this back to actually a circuit diagram, I'm afraid I'm going to have to, this is going to be a little tricky to use but I'll do my best. I'm used to working with a laser pointer. So here we have visual stimuli that are coming over here, and they are gated through the thalamus. There's the low road, the quick excitatory response to the amygdala. That's what makes your heart race and so forth. There's also the high road. This is going to the cortex, as we said. Thus far, we're still following the model presented by Joe Ledoux. But here's where it differs. So what's happening with your visual cortex when you experience something that frightens you is that your visual cortex is not processing all of the information at once. It's doing it in pieces, which is a very parsimonious and efficient way of handling a dangerous situation. If the danger is obvious, it will immediately pass through the visual cortex, process it only minimally, then go through the IFG. The IFG asks, do I have enough information to make a decision? If the answer is yes, it goes down to the ventral medial prefrontal cortex, which then suppresses the amygdala. So you go up, and then you respond back down. If the stimuli are more ambiguous, it might cycle more times through the visual cortex, this information loop, which we discovered, and thereby gathering more and more information until it's got enough processing through the visual cortex to then pass down through the amygdala, right? So in people who are trait anxious, and people who are naturally anxious, and even more so people who are have clinical anxiety, what seems to be happening is that this part comes down first, and then the IFG has such a high threshold that the answer to do I have enough information is always no. That basically they never have enough information through their visual processing to make a decision. And so this signal gets trapped here, which allows the excitatory response from the amygdala to dominate. In contrast, people who are at the what I would call reckless end of the spectrum, what's happening is that, again, they have this amygdala response. They come through here. As I said, this part of the brain seems to be atrophied. There's actually a part that seems to be missing. The signal immediately goes through and immediately suppresses the response. So it goes up, but it goes down immediately. So the assumption was always that people who take risks, they get high on those risks. There are adrenaline junkies, but that's not what we saw. We actually did measure adrenaline. We measured all of these hormones that are released when people are, presumably having fun. In fact, the people who were reckless, they weren't having more fun. They just didn't recognize the risk. So in fact, you might ask, so how do we sharpen the accuracy of threat detection? I'll go through this very quickly. We did this experiment. There was an experiment that showed that, that rats, when they're exposed to a predator, if you take that predator out and take the rat out and put a new rat in the same chamber, that the new rat actually freaks out, right? So this rat here sees the predator, freaks out. That's the technical term here. Take the rat away, take the predator away, put a new rat in here, and then he also freaks out. And so what's happening? What's happening is that the first rat is actually emitting an alarm pheromone through the anal glands, actually, which is then the second one is picking up on. So I was curious to know if humans did this as well. Now, notice that we were already kind of professionally in the business of scaring people. So it was an easy thing to do. What we had is a bunch of people. We had donors and detectors. So we had hundreds of people jumping out of planes, and we were collecting sweat in a way in which it was completely sterile. So you might think that sweat smells, but it's actually the bacteria that lives on sweat that smells. So if it's collected in a sterile manner, it's odorless. And so we had individuals donating this sweat. It was collected, pooled, and then presented via an olfactometer to a second independent group of individuals who were undergoing fMRI. So in fact, those people who were donors on one day were jumping out of a plane. The second day, at the same time of day for the same period of time, were running on a treadmill. So we didn't have to correct for genetic variants because we had both conditions from the same people. And in fact, the donors are in the scanner and they were breathing. Their instructions were to breathe in and out. They had these rings that were going in and out, and they must have been thinking, what a boring experiment. But in fact, part of the time they were being exposed to fear sweat, part of the time they were being exposed to exercise sweat, and part of the time they were being exposed to room air. They didn't know which because they were odorless, but their brains knew. And in fact, their brains selectively responded, the amygdala, which is the excitatory part of this control circuit, selectively responded to the alarm sweat, to the fear sweat, but not to the exercise sweat or to the room air. So this was kind of big news because it was the first time we'd shown that actually people can smell fear. And in fact, it was replicated, and it was even stronger the second time. And what we found, though, at the time, there was this guy kind of triggered a lot of fury in the media. Wired magazine said that I was constructing a fear bomb for the Pentagon. So, yeah. So in fact, if they'd actually stopped to read our paper, they would have noticed that was not the case at all. So what an alarm pheromone does is not make people, go wild with fear. I mean, this is not a very adaptive environmental response at all. The point of an alarm pheromone is to actually to communicate fear between animals of the same species, so that if I see a predator, if I scream, then of course the predator will find me. But if I emit a covert signal to my buddy over here, he knows that there's actually a predator about without detection. So obviously, if it just promotes mass hysteria, that's not a very adaptive biological function. What it does do is actually it triggers sharpening of threat detection. It doesn't make people see threat where it isn't there. It makes them more accurate in being able to tell the difference between threat and not threat. And we showed that both behaviorally and also in the brain. So there's a 43% increase. So moving on a little bit, I want to tell you actually about a project that we're just starting to do now, which is kind of taking this idea of personalized neurodiagnostics even more seriously and into the future, more ambitiously. And what we're trying to do is to understand what causes these circuits to self-organize. So these brain regions are talking to one another, but they could potentially talk to each other in many different ways, right? They self-organize according to different rules. One of them is obviously functional. So you saw that in a circuit they have different functions. Some are excitatory, some are inhibitory, there are filters and so forth, and they have to, computationally, the brain regions have to talk to one another in a particular order in order for the system to work. But there are also metabolic constraints, metabolic meaning energy. So the brain is about 2% of the mass of your body, but consumes about 20% of its fuel. And as a result, as such a fuel hog, it has to optimize over fuel, right? And so when it makes decisions about who to connect to, it's partially optimizing over the constraint of function, but also optimizing over the constraint of energy. And in cases where people have insufficient energy in the brain, and we hypothesize that this is occurring in people as they age, the brain starts connecting differently, we think, and that has implications for how people start thinking differently as they age. So we know, for example, that there is this hypometabolism. This is FDG-PET that I showed you. So this is a brain utilizing glucose normally in a normal, healthy person. This is the beginnings of mild cognitive impairment, and this is Alzheimer's disease. So what's happening is that the neurons are simply not able to utilize glucose. They go from being able to utilize glucose very efficiently, and remember, this is a fuel hog, to all of a sudden they're not being able to, actually all of a sudden is not correct, slowly, they start not being able to utilize glucose efficiently. And people had started to notice this epidemiological relationship between type 2 diabetes or insulin resistance and the cognitive impairment that starts to become visible with age. But I would say, the cautionary tale here is that most of these epidemiological studies have a really significant flaw, which is that when they try to identify the relationship between diet and something else, they're relying, first of all, on correlation, which correlation is not the same as causation, but they're also asking people to imagine what they ate, 10 years ago, 20 years ago. I mean, who can remember that far? It's not going to be very accurate. And so what we wanted to do was to ask this question in a much more precise way, to ask, how does the brain utilize fuel differently as we age, and what implication does that have for how the networks self-organize, and then computationally, how does that affect how people think? Now, the first point, if there are any neuroscientists in the room, the first very contentious point is that most neuroscientists, until very recently, and some still haven't gotten there, believe that insulin doesn't exist in the brain. In fact, this is a slide that is used to prepare medical students for their boards. And what you can clearly see is that the brain uses GLUT3, which is a glucose transporter that is insulin independent, unlike GLUT4, which is insulin dependent, right? So if, in fact, the brain doesn't utilize insulin, then someone can't necessarily be insulin resistant, right? That doesn't make sense. However, there are good reasons to believe that that assumption, and that, by the way, that medical school slide was from this past year, so someone needs to talk to them. There's increasing evidence that, in fact, that's a wrong assumption. So even as far back as 1998, it was shown that, in fact, there were insulin receptors in the brain. There was also this thing called insulin degrading enzyme, it's an enzyme that's meant to, as you might imagine, degrade enzymes, to remove it from the brain. If you don't have insulin in the brain, why would you have insulin receptors, and why would you have an enzyme that removes it? But most compellingly, here is a resting state study that showed reduced glucose uptake in insulin-resistant subjects versus healthy controls. So you can actually see that the brain uses glucose differently in individuals who are insulin resistant. So what that means is that neurons can, in fact, be insulin resistant. Now, we became interested in ketones because there was a study by Robert Sapolsky back in 1986. He was actually interested in something totally different. He was a stress researcher who was noticing that cortisol, which is a stress hormone, actually causes brain damage. But the way that it causes brain damage is that it actually makes neurons insulin resistant. And what he found is that if he just gave someone cortisol, that their brains would become damaged, and in fact the rats would become unable to perform cognitively. But in fact, if he gave them the same cortisol with also an exogenous ketone, the brain did not become damaged, and in fact they were able to perform task appropriately. So what that showed is that the brain has actually an alternate fuel source, that even when neurons are not able to utilize glucose, they might still be able to utilize ketones as sort of a backdoor to feed starving neurons. So here we see a group of papers by Tomasi and Volkow showing that initially, so long-range connections consume more energy than short-range connections in the brain. That makes sense because you've got axons that are connecting different areas. Obviously, you know, it's like a highway. If you have a longer highway, you have more to maintain. That's going to cost more energy. And in fact, the high consumption connections are the most vulnerable to aging. So these are the ones that start to degrade first. So if brain aging is a response to insulin resistance, tightening energy constraints, ketones should release those constraints. Now, does the brain optimize between function and energy when necessary, conserving energy by choosing metabolically cheaper pathways? So you can think about it this way. If you actually have to do a bunch of errands, so you need to get from point A to point B, and you need to actually do a bunch of errands, the path that you take would be different if you knew you were running low on gas, right? You might take a scenic route if you have enough gas, but if you don't have enough gas, you might actually have to take, you might have to go through a neighborhood, for example, that you'd rather not go through. So you have to make compromises when you're running low on energy. And so what we wanted to see is, in fact, do the neurons actually start to make those sorts of compromises when they're running low on energy? And is that then responsible for the cognitive difficulties that people have as they grow older? So here this seems to suggest yes. So again, this is the same group of individuals who are insulin resistant. And in fact, here they're performing a memory task. These are the healthy ones on top and the insulin-resistant ones on bottom. And you can see that it's not just less glucose metabolism, it's different glucose metabolism. The different parts of the brain are actually responding. That shows that the brain is rerouting. And we'd like to know how it's rerouting. So we can think about it this way. As I told you, we can start from a map analogy. We can say we have a destination. We have a certain amount of resources, and we want to get from point A to point B, and we're going to pick our route from many available options, right? So there are many ways I could get from point A to point B. We're going to use diffusion tensor imaging, tractography, structural imaging that I was telling you about to know which highways are available. So in the person's brain, we're measuring what the white matter pathways are. Those give you all the available highways that the brain could take in picking its paths. We have functional connectivity, which gives us which routes are actually taken, not just which routes could be taken, but which ones are taken. And then what we want to look at is the dynamic self-organization of networks, which is the interaction between getting to the destination you want to get to in light of limited resources, potentially. And so this is actually a problem that in statistical physics is very well understood. And so what we can do is have essentially an equation for the amount of gas available or ATP available, the route length or the ATP cost per route. We maximize caliber, which is just a fancy way of saying we use statistical physics to understand probabilistically what is the likelihood that given optimization over those first two, we pick one set of distributions versus another. So we pick one, we're more likely to pick this route versus this route. And then this will give us equations that will allow us to predict if we tighten constraints or we release them, how will this probabilistic distribution, this formation of networks change over time? And this will allow us to answer specific questions like, for example, if you have a network and we then tighten constraints on energy, is it the case that all of those connections just become weaker? Or is it the case that some connections that are expensive just drop out? Or is it that, in fact, that they completely restructure, that they use a different optimization strategy? And we think it's the latter. So here is what we're doing. This is a multi-scale model. So we're starting all the way from neurons here. These are leaky integrated fire neurons. All the way here, we're including metabolic pathways. So these are equations that give us glucose and ketone metabolism. Here we have the hemodynamic coupling, the neurovascular coupling, which gets us from the neurons to the hemodynamic response, which is measured by fMRI. Here is the engine behind it. This is the maximum caliber set of equations that I just told you, which does the optimization strategy. This produces brain-like signals, which then are making predictions. We test for individuals what those responses should be, and then compare. And if we are right, that shows us that our equations are correct, and if we're not right, then that gives us feedback that we then incorporate. So why would we have something like this? Because this is personalized medicine. The idea is not that we have 40 of this and 40 of that and do a statistical comparison. The idea is we take one individual, we put you in the scanner, develop a computational neuroscience model for your brain, and then can ask, how will you respond if I feed you this versus that? And how will you respond, not just now, or in 5 minutes or 10 minutes, but in fact, a year or 2 years or 5 years or 10 years from now. And so here is an example of this model. Maybe you could turn down the lights for 1 second. So here you can see that the neurons are firing. They're connecting to one another. And they're not firing in random ways. They're firing in ways that are actually anatomically constrained by real human data. And they're also firing in ways that are responding to changing metabolic constraints. Okay, so I think we can keep moving. There are clever ways to put this model together. You can't just simply willy-nilly put together whatever scales you want. In fact, there are specific places where the scales can talk to one another and specific places where the scales cannot, and so you have to pick strategically. What we're doing is asking again over periods of time, if we increase constraints, do the networks then have to switch their strategies? So in other words, I am setting out on my journey. I'm going from point A to point B. Now I tighten constraints and I don't have enough gas. Now, do I actually have to rethink my strategy? Do I have to rethink my route in that case? And so what we're looking at is how the networks self-organize and re-self-organize in response to these changing constraints. If they have to re-self-organize, then we know that they are picking a new strategy, and that leads to network switching in time. So here you have network switching over shorter periods of time and then over longer periods of time given by tau. So we have an experimental design where people are in the scanner, ultra-high field fMRI, MR PET. They actually run virtual reality mazes, so they're doing spatial navigation inside the scanner. And in one condition, they're given glucose, in the other, they're given ketones. So here we can look at what happens. Network stability degrades with age. So here are a bunch of data sets. One is with 600 subjects, one with 300. Here, this one is about with 20. So altogether, almost 1,000 subjects show that, in fact, what happens is that as we age, our networks start switching. So the networks, instead of being stable, when you're actually trying to perform a cognitive function, instead of your networks settling into a particular configuration and staying there, they actually are switching, which suggests that they're trying to re-optimize because they don't have enough energy. And what you see is that there's actually a clear switch point, which occurs around the age of 60. And that's true in both of these data sets, actually all three of these data sets. There's something very clear that happens around the age of 60. But in fact, if you look more closely, here over shorter time periods, you can see that even though short-term changes are occurring around the age of 60, long-term changes, holding a network online for longer periods of time, seems to be changing around the age of 45. Which is much earlier. So this suggests that there are actually this metabolic or energy constraints that are occurring much earlier than we thought, certainly before people become symptomatic at all. Interestingly enough, they seem to be stabilized by ketones. So here we have a glucogenic diet and a ketogenic diet, and you can see that the networks become stabilized, even though they become destabilized with age. Here in young, healthy individuals, these are individuals ages 20 to 38, the networks actually become like those of a younger person, even after being on this diet for just a week. Exogenous ketones amplify this effect even more. So this is an individual who's 44 years old, and you can see that the stability has already started to drift upwards but is brought right down to that of the younger individuals when you feed exogenous ketones. And this is true both for resting state, where they're just looking at an orienting cross, as well as while they're navigating a spatial navigation maze. So here, again, the network becomes stabilized into this spatial navigation network. And then it becomes, well, it's less stable than when you are with the ketones. So in other words, when people say that ketones are actually allowing them to focus more clearly, this is kind of what we're seeing in a neurobiological way. So does this degradation of network stability reflect a bias towards low energy states? It seems that it does. So the networks that were stabilized and destabilized were in fact the highest cost networks in terms of metabolics, so the ones that cost the most energy. And what we found is that the people with the lowest metabolism showed the most switching. So the networks with the lowest metabolism showed the most switching, which suggests that what the networks are trying to do is to find a better optimal strategy to having insufficient energy. So if I had to summarize, I'd say that personalized medicine requires moving beyond in several fundamental ways. First of all, it means moving beyond human brain mapping. So disease requires thinking about dynamic control processes, not just states, which is a fundamentally different way of thinking for medicine. Medicine's usually works by, you go and get a blood test and it's some particular value. And that value is too high or it's too low. And we have to get away from that way of thinking because that's actually not the way that you really would diagnose a disease process. So from that perspective, we need philosophers because this is a conceptual change, not simply a scientific one. Second of all, we need to move away from a reliance on statistics. So there's nothing wrong with statistics except statistics assumes linear relationships that are not appropriate for control circuits. And we said that most of physiology and our conception of disease depends upon control circuits which are nonlinear and therefore not really describable by statistics. So we need other tools. And those tools do exist in other fields. So coupled differential equations, dynamical systems, chaos theory. So in other words, we need an influx of theoretical physicists who can bring to bear those tools. And third, we need to move away from the idea that more data is the way to go. So it's not bad to collect more data. I mean, it's important that we are now pooling resources and making use of these large shared databases. But at the same time, it's not getting us necessarily closer to our goal of personalized medicine, which is fundamentally a process of looking at a single individual. So we need to think about acquisition parameters for instrumentation that are optimized specifically for a single individual rather than large groups. So from that perspective, we need engineers. So thank you for your attention. Yes?
I would just like to hear more about the ketone diet.
So it was, this is a low carbohydrate, medium protein, high fat diet that the subjects were put on for a week. They were given a set of instructions and they were told to do originally urine tests at home to figure out when their ketone levels achieved our requirements, and then when they came, we did a blood test just to confirm.
One more question.
Sir?
That was brilliant.
Thank you.
I now understand 40 years later why I jumped out of airplanes and why I quit. But thank you so much.
That wasn't really a question. Okay.